All right, thank you. So as an evolutionary biologist, this is going to get loud and quiet. I'll try to stay still. I'm a walker, but I'll try to stay still. As an evolutionary biologist, I'm really interested in the mechanisms that maintain variation within populations. And this isn't a unique interest. In fact, this has a long history in evolutionary biology, is a key component of the modern synthesis, as I highlight in volume 4 of one of Sewell Wright's series on the evolution and genetics of populations, was about the maintenance of variation within populations. And since then, there's been a rich theoretical and empirical literature that is built around how variation is maintained. And one of the reasons why there's this history of studying this is because it poses a bit of a conundrum, especially for traits that should be under selection. There really should be an erosion of variation in those fitness-related traits. So what is maintaining the variation? So some of the mechanisms that have been proposed as being important in maintaining variation within populations are things like spatial and temporal heterogeneity in selection, balancing selection, frequency-dependent selection, bet hedging, and then non-selective processes like gene flow and genetic drift. And obviously, there's an element of uncertainty and stochasticity with gene flow and genetic drift. But in fact, of the selective processes, most of those have to do with mitigating or dealing with uncertainty in the environment. And so it's really this uncertainty that is responsible for maintaining variation within populations. So I'm interested in exploring a little bit more about what is the role of uncertainty in this context, and specifically that neither the biotic nor abiotic environment are fully predictable. And so really what organisms have to do is find ways to mitigate or buffer against the uncertainty. And we've heard some of this in talks today. Almost all of those were about variation along a gradient or a geographical range and how different populations might be responding differently. What I want to talk about more is that unpredictability within a population. And this is especially true for traits that are closely tied to fitness, that really being able to deal with that uncertainty is crucial. So if we think of just a basic annual plant with a very easy sort of to understand life cycle, there are a few key aspects here where plants have evolved strategies to integrate cues from their environment so that they can deal with the unpredictability of the environment, either within a season or between seasons or at a small spatial scale. So some of those cues have to do with temperature and photoperiod. So for example, the timing of when seeds germinate, whether they have dormancy, so that they can time the appropriate season, whether that's a fall germination or a spring germination. Integrating cues about temperature and photoperiod for when you flower. And then also kind of when you set your seed and having that next step be dependent on the step before. So there are a few things here about dealing with the environment. The other thing is that how you mitigate that uncertainty and when you go through those life transitions. So for example, when your seed germinates, sets up the environment for the next life stage. So that small rosette is either you're either going through winter as a small rosette or a seed, or you're going through spring in that stage. And when you're maturing your seed is conditioned upon when you flower, which may be conditioned on when you germinate. So there's multiple kind of conditions here that is mitigated by the step before and correctly integrating those cues. So what I'm going to focus on for this talk is this germination difference that I've highlighted here and how populations might use their the variation in their environment to determine when to germinate, and then the effects that might have on later life stages. So in this photograph here, I show what fall and spring germinants look like. This is in Mimulus guttatus, which I'll get to in a minute. But these photographs were taken on April 4th in a single population on Vancouver Island in British Columbia, where we have new spring germinants as these tiny little cotyledons. And we have some larger rosettes that germinated in the fall and overwintered as a rosette. And we were interested in understanding why both these strategies exist in the population. So if there's a genetic basis to the seasonal germination or dormancy, and then what effects this might have. So if you're coming out of spring as this larger rosette, what effect does that have on your life cycle? And sort of what's underlying this difference here? So are there fitness consequences for these different germination? And then is there any effect on the next generation, perhaps because those parents experience different environments? So if they flowered at different times or had different resources to invest in seed, or the seed experienced different temperatures or photoperiods, would that have any effect on the next generation? And all of this work was done primarily by a fantastic honors undergraduate student in my lab, Taylor Middleton, shown here in front of a poster on this work. And she was largely mentored and supervised by my postdoc, Matthew Rubin. Okay, so this is the population that I mentioned on Vancouver Island in British Columbia. This is what it looks like in early April when those photographs were taken. And this is what it looks like at the end of flowering in June. So this is a very short life cycle for this annual population. So that's kind of a zoomed out image of all those carpet of early spring germinants, and then later in the summer when they're all flowering. So in an ideal world, Matthew and Taylor would have camped out at the site for a year and measured and tagged every single seed as it germinated in the fall, when it came out from under the snow, been there that day, and recorded everything. Unfortunately, that wasn't possible. So we did what we considered the next best thing, was to simulate the environment at the site in a growth chamber. So we downloaded data from an environmental station that we had there, because we had a field site for a different experiment there. And we had the growth chamber run a dynamic program that reflected the timing of sunrise, temperature, and humidity throughout the 6-month period. So we started our experiment when the rains come and the seed germinate, the start of October, and ran it through till late spring when drought sets in and everything senesces. So we used 20 maternal families, and essentially we're looking at these two different germination cohorts. Okay, so I'm just going to get right into the results here. So first, I'm showing you here the distribution of the timing of germination. So this is our experiment day. And you can see fall plants germinate, and then a while later, spring plants germinate. We accelerated winter a little bit here. And just to point out that the distributions of germination are actually quite similar. Really high germination in both fall and spring. So in a single population, seeds are germinating in either season. Here I show the means of our maternal families and a reaction norm of them across the two seasons. And except for this one family that showed strong cold-induced dormancy, there's actually very little difference. So we're not finding genetic variation in the timing of germination. The seed are basically germinating in equal proportions in both fall and spring, except for one family that I'll come back to later. Okay, so this was the first result we didn't expect, but that's what we got, so we're going to go with it. So seed are essentially germinating whenever, and then we wanted to look at what are the downstream consequences of this for the next life stage. Well, we found that there is a difference in the onset of flowering, or the start of flowering, but that they do overlap. So the fall plants essentially start flowering here, and about 20 to 25 days later, or that's probably about 15 days later, the spring plants start flowering. But these keep flowering. So for most of the flowering period, the fall and the spring cohorts are overlapping. So they do not have separate flowering times where there might be assortative mating within cohorts. So there's the potential for full mating pollination between the fall and spring germinants. So the flowering overlapped. What we did find, so there was a difference in flowering onset. So I'm just flipping those distributions on their side now to contrast the differences here. So the flowering onset day differed significantly for fall and spring. So that's sort of the day of the year that they start flowering. And what this meant was that there was a difference in flowering duration. So fall plants essentially keep flowering for longer than spring plants. And the reason why fall plants flower for longer is actually not necessarily because they have lot more resources to invest in growth and flowering. The reason why they flower for longer is because death is highly synchronized. So the end of the growing season happens for all plants all at once when drought sets in and the rains stop. And so the fall plants have a longer growing season, have a longer flowering season because they start flowering earlier. All right, so they have a longer flowering season. What does this mean for fitness? So timing of flowering is not actually necessarily a good proxy of fitness. So we wanted to know for this annual plant, how many flowers do they make? The fall and spring cohorts made exactly the same number of flowers. So this was really surprising to us. So despite being much larger when they come out of the snow, as I showed you in those early photographs, flowering for significantly longer, they don't make any more flowers. So it really looks like there's no benefit in terms of reproductive fitness for flowering earlier or for flowering for longer. They're sort of maxed out in how many flowers they can make. So one question you may be having is how those spring plants caught up. How were they able to make the same flower number when there were those tiny little cotyledons and the other ones were those nice big rosettes? Well, it turns out that the fall and spring cohorts actually have different growth strategies. So the fall plants have this longer growing season. They're making many more leaves, becoming a larger vegetative rosette, but each of those leaves is actually smaller. And we think that rather than investing in the expensive construction costs of making more leaves, the spring plants are making fewer leaves, typically only 4, so two nodes of leaves, but making larger leaves. So they're rapidly expanding their leaf size rather than making additional leaves, giving them enough resources to set the equivalent, to make the same number of flowers and set the equivalent number of seed. Okay, so one question we had following on of this is whether those seed differ in viability. So it's not just about how many flowers you make and how many seed you make, but really the consequences of those seed. So they may differ for 2 reasons. One might be because their parents experienced a different environment. The other might be if they invested differently because they had different resources. So if those fall plants still were bigger, did they invest more? So we did a massive amount of crossing where we crossed all potential parents in all directions so that we could account for maternal and paternal effects in germination. And so we crossed reciprocally within fall, within spring, between fall, between spring. We knew the maternal family of all these plants, so we controlled for that. We then grew up seed in 2 different environments, with and without cold, to see if the seed had any preference for whether they germinated in the fall, no cold, or the spring cold. We accelerated this now. And we just did these in plates because we had 8,000 plants and no growth chamber space to accommodate that. All right, so what did we find in terms of the progeny? Well, so this shows essentially fall and spring, so no cold, cold. Germination proportion. You might look at this and see some really cool crossing reaction norms. Back out a second and look at our scale here is 0.98 to 1. Absolute. I was going to have that on a 0 to 1 scale, but it actually looked stupid. So there's absolutely no difference in germination. They all germinate at basically 100%. 100% viability for this nice growth chamber seed. The days to germination accelerated with cold, which is a really common pattern, but had nothing to do with parental effects. So no contingency from the parent population to the offspring population in this somewhat contrived growth chamber experiment. Okay, so really we're finding that the timing of germination doesn't really matter. These annual plants have a growth strategy that gets them to equivalent fitness. I just want to briefly come back to this one population. that did have a cold-induced dormancy. So we had offspring from this in our crosses. And so the offspring, again, had 0 germination in the cold if they had one of those plants from this family as their parent. My postdoc is following up with this. Through some basic crossing and growing up progeny, we think that it's a dominant mutation. It's cold-induced. We think it's a single gene, but that might change, so I'm not going to say anything about that. But it's a nice example of there might be some variation within the single population, but most of it is about they just deal with the environment that they're given. So to summarize, the variable life cycle based on germination timing exposes plants to different environments. They are really different in size when they come out of winter, and that really affects growth strategy. So it's a plastic growth strategy response to deal with when they germinated that then gets them to equivalent fitness. And we think that this variation in germination in the field is just driven by stochastic availability of water. So if you're a seed that lands in a water patch, you germinate right away. If you don't, you wait through winter, rains come in spring, you germinate in spring, and then you're conditioned to grow differently and flower together and equivalently. So I just want to briefly mention, because I don't want you to go away thinking that cold doesn't matter for Mimulus guttatus, because we've previously shown that things are really different for perennials. So in a previous paper, we had shown that annuals, the red line here, flower regardless of cold. So this is now across the entire range of Mimulus. Annuals flower regardless of cold. Perennials flower in lower proportions if the seed don't get cold. And we similarly have shown that annual populations don't need vernalization to flower, while perennials do. And really what we're showing is that annuals have this really short life cycle where they have one shot at fitness. And so no matter the environment they come in at, whether it's before cold, after cold, they just flower and they give it their all. While perennials might have more flexible strategies where they need cold at either the seed stage or the rosette stage to time their flowering. So to wrap up here, I just want to highlight the importance of studying variable traits within single populations. So a bit of a different perspective from some of the talks we heard from this morning, because it really provides an opportunity to test fundamental mechanisms of evolution, but that mechanisms of mitigating uncertainty might vary, so there's different ways of doing this, and even within a single species, if we contrast the annuals and perennials, it seems like they're dealing with the uncertainty of winter in different ways. Thank you.